Okay, so today, guys, we're going to talk about DNA and protein synthesis. And we're going to review a little bit of some very important concepts that I want you guys to have before you're able to actually get the whole concept of protein synthesis. By now, you should know what the structure of DNA and what happens during DNA replication. So we'll go and review that too. I'm going to skip through the history, and you can look up, well, we'll go over a little bit of it. Erwin Chargaff discovered the base pairing rules and ratios for different species. Adenine pairs with thymine, and cytosine pairs with guanine. Rosalind Franklin and Maurice Wilkins had taken the first pictures of DNA using X-ray crystallization. This proved that DNA had a helical shape. And Watson and Crick became a historical footnote and were known as the fathers of DNA because they actually took this information and they pinpointed and named the actual structure of DNA, and they call this the double helix. And the double helix it is actually just this, and this is, kind of a cartoon to make fun of it. As you can see from the model, the structure is basically a long, twisty rope laddery type thingy. And the double helix is just two strands of DNA, and it's twisted and it's turned, and it does this through hydrogen bonding, which we've already talked about up to this point. And what you get is a double helix. One thing that I like to show my students is this guy doing what is called the double helix, and as you can see, it's just a goofy dance, but hopefully that will help you to remember that the shape of DNA is double helix. By this point, you should know that nucleic acids are made up of nucleotides, and nucleic acids are their function is to store and transmit genetic info. Examples are RNA and DNA. So when I ask you, DNA is an example of which type of macromolecule, you should probably know that it's a nucleic acid. And you should also know that nucleic acids are made up of nucleotides. And that's kind of easy to remember because nucleic acids, nucleotides, they've got part of the word inside the monomer. And you should know by now that proteins are made up of amino acids. So if you're asked anything about the makeup of proteins, you should know that they're made up of amino acids. And proteins have, and that should say, many different functions, and we will go over most of them in a few minutes. Just to give you kind of an overview of DNA, we've talked about chromosomes, we've talked about mitosis, we've talked about the division of the cell. When we're looking at a chromosome, this is actually, if we unwind it, this is DNA wound around proteins, okay? And it's actually taking the DNA and it's wound around these proteins, and these proteins are condensed together and they form a chromosome. If we take DNA and we unravel it from the chromosome and we look at it, there are segments on this DNA that are called genes. And genes are going to code for traits. And I'm sure previously you heard the word gene before, and you know that genes are what make us who we are. Or you've heard the expression, you have good genes, or you've got your mom's genes or your dad's genes, and that just comes from this. We inherit our DNA from our parents, and on this DNA we have something called a gene. Okay, so I'm going to keep going, and we're going to talk more about genes next week and the week after that. So here we have DNA, and DNA is made up of 3 parts. It's made up of a phosphate group, a sugar, which is deoxyribose sugar, and it's made up of a nitrogen base. DNA actually stands for deoxyribose nucleic acid. So that should help you remember that it is a nucleic acid, and that its sugar is deoxyribose. And if you ever want to sound, smart in a situation, just whip that word out, deoxyribose nucleic acid. You can just say it, just deoxyribose nucleic acid, and instantly you're going to get some street credit. But here we have a phosphate group is also part of it, a sugar base, and nitrogen base. And this nitrogen base can be one of 4. The 4 nitrogen bases are adenine, thymine, guanine, and cytosine. And up until this point, you should have learned that adenine binds with thymine, and cytosine binds with guanine. If you're ever in doubt of these, you should know there's one phrase that I like to tell my students to remember: the 4 nitrogen bases, and that's all tigers cry grr. All tigers cry grr. Adenine, thymine, cytosine, guanine. And A is going to bind with T, and C is going to bind with G. And just, you should just say that now. A binds with T, C binds with G. There are 2 types of nitrogen bases. We have the double ring, which is guanine and adenine. Those are purines. And if you look at this, you're seeing a single ring. So there would be another ring out here. That would be a double ring. And then we have the single ring, which are the pyrimidines. And that's cytosine, uracil, and thymine. And up until this point, you should have heard about uracil and know that that's not actually found in DNA, but that's found in RNA. And move on. And a way you can remember the purines is PGA, purines, guanine, adenine. And then a way to remember the pyrimidines is cut pi. And here we have DNA's double helix, okay? If we untwisted it and looked at it, then, and we took all the twisting out and it was just this flat structure, it would look a little bit like this. And look, if we look at it closely, it reminds us of a ladder, okay? We have the rungs of the ladder, which are nitrogen bases. And we have the sides of the ladder, which are the sugar bases and the phosphate groups. Okay, so you have sugar bases, phosphate groups, and binding these binding these nitrogen bases together are the hydrogen bonding. So we have hydrogen bonding these guys together. And here we have the purines, which is double ring, and the pyrimidines, which is a single ring. And I just kind of want to show you guys, if we kind of just look right here, that is a nucleotide right there. There's a nucleotide. Here we have our nitrogen base. Here we have our sugar base, and here we have our phosphate group. And actually, I kind of want to look at it like this. If we pull that apart. Okay, moving on. DNA is the genetic code, is the sequence of DNA nucleotides in the nucleus of the cells. And pretty much what that says is just what I was talking to you about genes, okay? We pass genes on to our parents, or I'm sorry, we pass genes on to our offspring. Parents pass them to offspring. And these genes are actually made up of sequences of nucleotides. So we have different sequences of nucleotides, and those different sequences of nucleotides are going to actually control whether we get blue eyes or green eyes or brown eyes, and just the sequences of DNA, the different sequences. So DNA is a double-stranded molecule, and you should know that by now. we have one strand bonded to another strand. The strands are connected by complementary nucleotide pairs, and all that means is adenine bonds with thymine and cytosine bonds with guanine. And they have the backbone or the sides of the ladder that are made up of deoxyribose and phosphate groups. And we have the rungs of the ladder, which are made up of nitrogen bases. And what bonds these nitrogen bases are hydrogen bonding, okay? And if you look closely, between thymine and adenine, we're going to have 2 hydrogen bonds, but between guanine and cytosine, you're going to have 3 hydrogen bonds. That might be a nitpicky question that they ask you on the EOC, but that won't be anything I ask you on tests. The ladder twists to form a double helix, and actually the way it twists and forms is also, again, through hydrogen bonding. DNA is actually copied during the S phase or S stage of interphase. And DNA just makes a copy of itself. And I taught you guys, or you guys watched a video on this earlier, and pretty much we need 2 copies of DNA because the cell is about to divide. Okay, so there needs to be 2 identical copies of the DNA for the cell to divide, and it makes a copy of itself during S phase, and that happens in interphase, the resting period of the cell, or the resting period of the cell cycle. DNA is called a semi-conservative process, and you guys should have already gone over this before. It's a semi-conservative process, and semi-conservative means that you conserve part of the original structure in the new one. Okay, so here's our parent cell, and it's purple, and here we have our new daughter strand being built onto the parent strand, which is purple, and the daughter strand is red. And what happens is, and I've talked to you guys about this before, is the parent cell splits in two through an enzyme called DNA helicase. DNA helicase actually splits that parent cell right here. It splits it, and then it uses DNA polymerase to build a new daughter strand onto the existing parent strand. Okay, and what happens is we end up with 2 identical strands, and half of that strand will be part of the original parent strand, and the other half will be part of the new daughter strand. Or half of it is the new strands of DNA. Half of it is the parent cell or strand, and half of it is a daughter strand or a new strand that's been built on. Okay, DNA is a gene. A gene is a segment of DNA that codes for a protein, which in turn codes for a trait like skin tone or eye color. And we'll learn later on that skin tone and eye color, we actually have many different genes controlling that. But just to talk about, DNA, a segment of the DNA is a gene, and that segment or that gene is actually what helps us build proteins, okay? And these proteins actually are what make up that skin color or that eye color. We actually have a protein called melanin, and that is what makes up our skin tone, okay? And this gene right here could very well be the gene that codes for melanin. A gene is a stretch of DNA. So, just a little review. DNA contains genes, which are sequences of nucleotides, and these genes code for proteins, okay? And what that actually means we'll get into a little further, but you should know that the genes actually tell the cell what proteins to build. And proteins are used to build cells and do much of the work inside the cells. So here's a review of proteins. Proteins are made of amino acids. We just said that. They're linked together by polypeptide,